welcome back to the second half of our semester. Previously, in the first half, what we focused on was pluripotency. So we mainly discussed how you can derive pluripotent stem cells. Basically, we have three ways. One is the natural way. You can mated animals and then isolated blastocyst. Then you can have you will have embryonic stem cells. That's the gold standard of pluripotency. And then we have two methods of reprogramming. One's through oocyte, enucleated oocyte. In the cytoplasm of oocyte, it possesses the power to reprogram a somatic cell nucleus. And then based on the work in somatic cell nuclear transfer, over many years, scientists eventually were able to just put in some transcription factors or even chemicals into somatic cells, and then the somatic cell will experience a long process that is not fully understood at the point it eventually become potent stem cells. So that was the major focus of the first half of our semester. Is mainly about pluripotent stem cell. Apparently, pluripotent stem cell is super important. That's why we put half a semester's of effort in studying that. So why it is important? First of all, it can be used in tissue engineering. So when you have a when you have a pluripotent stem cell, theoretically you can derive any cell, somatic cells, out of pluripotent stem cell. And secondly, pluripotent stem cell is a good model to study differentiation or development because it can give rise to any cell type theoretically. So we can study how individual somatic cell is, how they are derived from the embryonic stem cell. Usually it takes many steps. First of all, it's going to become a certain germ layer and then become some kind of progenitor and another progenitor. Many steps. Eventually, it will give rise to a somatic cell type. So in the second half of stem cell biology, we're going to focus on adult stem cells. And now we jump to individual organs, such as the heart, the neural system, and others. We describe the adult stem cells, how they contribute to tissue repair, regeneration, and so on. So it's more about application, but as you have realized already, so we are more interested in the pathways, the regulatory mechanisms, not the eventual outcome. So although ultimately we want to use our knowledge to treat diseases, but that's usually not the task of basic scientists. So the clinical doctors, the industry eventually will try to refine a strategy that scientists developed and proposed to them. They're going to refine the technology. The clinical doctors are going to have a large sample size to see if therapy prototype is indeed feasible in humans. But for us, we don't have that burden. That's what we're good at. So we are studying the mechanism of action, MOA. Like I said, even if you show in animals that thing worked so efficiently, like you put some therapy to the animal, you achieve the outcome you wanted to achieve 100%, that's not enough to convince others that this is a good therapy. You may wonder why, it is so convincing based on that observation alone. The reason is because the cause and outcome is hard to establish, especially when in biology you often deal with a limited sample number. Like if in a lab, you probably can analyze hundreds of animals, right? At a given period of time, that's hardly sufficient to establish a causal role of a certain process. So you need to, but once you start to appreciate the fundamental mechanism, and then things will become clear and clear. And then the outcome will be natural, will be very natural. So that's why if you propose something to the pharmaceutical industry, you say you have something, they will first, of course, will examine what you proposed, the outcome. If the outcome is exciting enough, and then the next question they ask is going to be, what's the mechanism? If you don't have the mechanism, well, they will not be interested in. No matter how exciting your result is, the outcome is, it doesn't matter that much to them. Okay, so as I said, we're always going to focus on the fundamental side of stem cell biology. In today's lecture, I'm going to describe regeneration. So what is regeneration? So that is a term closely related to stem cells, because people feel no matter it is a pluripotent stem cell or adult stem cell, the function of these cells is to replace the damaged or diseased tissue throughout the life of the animal. So it's a pool of cells there. Usually they don't have a function, but if it's the individual organ or tissue is injured, and now the stem cells will jump out and start to do their job. So you know this term, regenerative medicine, therefore is closely related to stem cell biology. So there are basically three strategies, that you can easily think of that can be applicable in disease settings. The first will be direct inject of a stem cell population. If you know, there are a stem cell population in the heart, and then a very straightforward thinking will be, if the heart has some injury, you just inject this heart-specific stem cells. I mean, these stem cells will automatically differentiate and then repair the damage. So that's a very straightforward way of thinking, and apparently you can try that in therapeutic settings as well. And next, you can kind of lure the cells toward a certain fate, and then inject those cells for certain disease settings. So the reason is because the stem cells will have different levels of potency. Some are pluripotent, some are multipotent, and unipotent. So if they're multipotent, they can differentiate into many different cells. In a disease setting, you may not want the cells to differentiate toward all the cell types that the stem cell is capable of. It may not give the advantage. For example, if a stem cell can give rise to endothelial cell, smooth muscle cell, and a skeletal muscle cell, in one situation, maybe you only want endothelial cells. You don't want the other muscle cell types in there. It could interfere with the repairing process. So in some settings, you can lure the cells toward a fate. Once that commitment has started, now you inject the cell. So that's the second strategy. So instead of injecting the stem cell, the original stem cell, you first differentiate the cell somewhat and then do the injection. That's the second strategy. And third will be like inducing the endogenous stem cells without isolation. Using heart as the example again, if in the heart you have some stem cells, during some kind of a disease process, maybe you can inject some drugs that can induce the stem cells now to differentiate and then repair the damage. So there are the three ways shown on this slide that are the very fundamental strategies that one can predict that may be used in therapeutic settings. But one very important concept here is you don't always need the stem cells for tissue regeneration. So in today's lecture, we're going to first describe some settings in which you do have stem cell involvement. And in the next lecture, I'm going to describe some processes in which you don't have the involvement of stem cells, but still you can have regeneration. Okay, so I hope you can still remember the definition of stem cell. To be a stem cell, you need two criteria. First, it must be able to self-renew. Second, it should be able to differentiate toward other somatic cells. So to involve stem cells or not, you need to figure out in that process, do you have these stem cells as we have defined involved in that repairing process? Okay, so regeneration doesn't always involve stem cells, no matter it's pluripotent stem cell or embryonic or adult stem cell. And shown on this slide, you also see this beautiful creature. Can somebody name this creature? Somebody want to try what this is?
Salamander or something?
Very, very good, very good. This is a I believe this is also salamander. I don't know just how you define salamander, but usually this is called a newt, N-E-W-T, a newt, or this is also called Urodila. It's called Urodila as well. That's the two names for it. This is not a lizard, although it looks like a lizard. It is not a lizard. It's an amphibian, just like frog. But for frog, usually only the young individual will have tail. But for a newt, even the adult will still keep the tail. Okay, so
Professor,
yes.
Sorry to interrupt you, but some reason I cannot see the screen. I'm not sure, is it me or is it?
I can see the screen.
Oh,
Yeah.
yes, and then
Yeah.
it's me.
Can you restart your team?
Yeah,
See
please.
if it helps, or the computer see if it will help. Yeah, it's a newt shown there. And today I'm going to show you quite a few different species. I hope you can kind of memorize the names. First of all, they're just wonderful creatures. Better remember those names. And then secondly, they are the models used in regeneration study. And this is beautiful work. I would say it's much better than mouse-based work, because either in mouse or in humans, the regeneration power is not as great as in lower species. So if you really want to study regeneration, you probably will end up with studying this lower species. So why don't you just remember their names? So this is a newt here. And on this slide, I want to describe to you one general rule or summary of the regenerative ability of different animals. Actually, it is very hard to provide a general principle of the regenerative ability, because some lower species have great regenerative ability, but not always. I'll explain to you why. But please look at that figure in the middle of the slide, and here on the horizontal axis, what you see is increasing regenerative ability, decreasing regenerative ability. Well, on the vertical dimension there, it's the time of evolution. So the humans apparently are at the top of that vertical dimension, because we're the latest in terms of generation. So we start with invertebrate. There are a few animals listed here. The first one, I hope you can see my arrow. That is called a flatworm or planarian. So that's a planarian there, the yellowish ones, the flatworm. And then the next one is called hydra. And the third one is a starfish. And then the next two that collectively, I couldn't remember it's called anthropod. Collectively, they are called anthropod. That include crustaceans and insects. They're collectively called anthropods. Basically, they have many legs, okay, like a crab and lobster. Crab and lobster are crustaceans, and then the last one over there is an insect. So all this shown on the first column are invertebrate. And some of these have really, really great regenerative power, such as this flatworm. The flatworm is kind of immortal. They will never die. You can cut them to pieces. The more pieces you cut them into, that means the more individuals you're going to get. Each pieces will give rise to a complete new individual. So that's the amazing regenerative power there for some species. Amazing. Because before I teach this course, I didn't know there are such creatures in existence. That's, for example, like even bacteria. If you break the bacteria to two parts, they are not going to recover. They are not going to give you another bacteria. But these creatures, this flatworm and hydra, the next one listed here, they both have the capability to just completely regenerate themselves from just a piece of the original individual. So, and next, this crab and insect and some of the vertebrate, like showing in the middle part of this slide, we have that the first creature in the middle part, that is called axolotl. The axolotl shown there. And then you have a tadpole here. The grayish one is tadpole, it's another amphibian. And then the third one is a zebrafish. So these are vertebrate, but lower vertebrate. And this can also regenerate some part of their body. For example, the axolotl will be able to regenerate the limb. So if one of the limb is removed somehow, so sometimes they just, when the predator pull their leg, they will just remove the leg and then escape. But with a few days, usually about a month to 2 months, they can regenerate that limb completely. And then the zebrafish can regenerate the fin. And tadpole or other frogs, they can regenerate their limbs as well. So as you can see, some of the extremely lower species like flatworm can regenerate completely, while some lower invertebrate can generate a structure such as the limb. But once you move on to higher vertebrate, like humans and mouse, rodents, and that deer, and now the regeneration power has reduced dramatically. For us, for example, maybe we can regenerate our skin. Like if you have some part of the skin, I would say maybe the superficial level layer. If every day maybe some cells get regenerated. The lining of the intestine get regenerated every few days because the intestine is always exposed to toxin in the food, things like that. So the epithelial cell layer get replaced quite frequently. And then if you get a COVID infection, maybe some lung cells will die, and then they eventually get replaced, regenerated. So we still have some power, but compared to the lower species there, the power is much limited. Same for mouse, and for that deer, the antler can be regenerated. Okay, so the general principle here, we say by and large, the capacity to regenerate is observed less in complex multicellular animals, yet in the lower species, the power is greater. That's just a very very raw principle there. Why we say that is because even in the lower species, amazingly, some of the close cousins of those species doesn't have the same level of regeneration power. For example, like a flatworm, that particular flatworm called planaria have the total power of regeneration, but some species that are really closely related to it don't have the same degree of regeneration. So we don't know why, but that provided scientists a great resource for us to study regeneration, because when you do a comparison, you want to have a control group and then to study whether your test group have the effect that you want to observe. The best is that control group is close enough, right? Like in humans, if you're studying two individuals, if the two individuals are twins, and then that comparison makes a lot more sense. You're likely to find something that's real. But if you compare two people from very different ethnic group, and when their ages are different, apparently your conclusion are not going to be as solid, right? So when in nature you have closely related species, one have great regenerative power, while the other don't have as great regenerative power. Well, that's a great system for you to study regeneration. That's exactly what scientists are doing. Okay, so the general principle is very, very general. Okay, so it won't apply when you get into details of regeneration. So on this slide, I'm going to talk about the different level of regeneration. Just because we're going to discuss regeneration, I want you to kind of appreciate the different levels. So again, you can see shown there is the planarian, and the planarian flatworm. By the name, they're worm, just kind of flat. So they can regenerate the whole body. So shown on that slide, you can do an amputation anywhere. Basically, you cut an animal to several pieces. In this example, you cut the head, you cut the tail, you only leave the middle part there. But that middle part is going to regrow the head and regrow the tail. So that the new individual will be formed. So that's a total regeneration. And then next is going to be the regeneration of a structure. So the most widely and best studied will be the regeneration of limb in amphibians. Okay, so the scientists usually will amputate a limb from an amphibian and then study the regeneration. So we're going to have some description of the work done by some well-established famous labs doing that research. And for this Friday's student-led discussion, they are going to describe limb regeneration as well, because that is the best studied model system for regeneration. And the next level will come to more limited structures, such as internal organ, for example, like heart. So in this lecture, in next lecture, we're going to have a very brief discussion of heart regeneration in zebrafish. So basically, if you amputate the apex of the heart, zebrafish will be able to regenerate that part, but only a small portion of the heart. If you cut the heart to two pieces, there's no way the full function will be regained. So you can tell that's a loss of regenerative capability when you move up of evolution, and when you compare adult animal to a much younger animal or a fetus. So usually in adult, the regenerative power is less. So that's another general principle of regeneration. Lastly, tissue regeneration occurs even in humans, like the epidermis and the gut lining, as I discussed. The intestine lining gets replaced quite frequently. You can see, different levels of regeneration as that will be the content we discuss in today's lecture. Okay, I would like to introduce this very important term that set models, good model systems for the study of regeneration. So this term is called autonomy. So what is autonomy? So autonomy is a term that is used to describe a process of regeneration. The term itself basically means amputation, a self-amputation. So many animals do that. In our backyard, you may have some lizard. You see some lizard. If you try to catch them, they can shed their tail. They just drop the tail because they can regrow it back, right? That's a very good example of autonomy. So the animal intentionally remove that part of their body so that they can avoid getting captured by their predators. So autonomy basically means self-amputation. So this phenomena, is of course occurred naturally in the animal world, but scientists noticed that, and then animals who does autonomy, get to become really good model systems in the field of regeneration. So we have a few examples, such as the autonomy of limbs and tails in salamander and lizards, right? So one of the salamander will be the one I showed in the previous slide. So that, I'm sorry. So this axolotl, this thing is a salamander. Okay, so it's called a Mexican salamander, I believe. So the salamander and lizards can remove their limbs and tails. I'm sorry, salamander can remove their limbs, and the lizard usually will remove their tails if they face a dangerous situation. So those are lower vertebrate, right? So a very interesting observation is that in higher mammals, such as mouse, here we show a mouse called African spinning mouse, or Acomis is the name for this mouse. They can autotomize their skin. That is very strange. So when they face danger, they get captured by their predators. They can remove a big chunk of their skin and then escape. So that's probably the only case of higher mammals remove a lot of their body parts, as a term of autonomy. That's probably the only example for higher mammals. Otherwise, it occurs in lower vertebrate, such as salamander and lizard, and of course, it will occur in much lower invertebrates, such as crustacean and insects, right? And of course, in even lower ones, such as the starfish. Here is a sunflower starfish. You can see the one arm is now autotomized. It's removed the one of the arms to avoid being captured. And then this is a lizard, and the third one is a mouse, the African spinning mouse. The skinny mouse can remove the skin as a process of autonomy. Okay, So that's an important concept there called autonomy. The animals who can do autonomy are good model system to study regeneration. So on this slide, I want to describe several important concepts. In this lecture, we have many terms. That could be very useful for your future study of if you are interested in regeneration of lower species. Try to understand the meaning of those terms. Okay, so this slide has three main concepts here. One is called differentiation, which is easy. Differentiation means a stem cell will become a somatic cell, become more specified. That's differentiation. But we do have two additional terms related to differentiation. One is called de-differentiation, with a DE before differentiation. De-differentiation. The third is called transdifferentiation. So three terms here. I will explain them. And also on this slide, another major concept is that regeneration doesn't always include the involvement of stem cell. So observe the three processes shown here. So we start with a fertilized egg. That's the beginning of life. This fertilized egg eventually will give rise to adult stem cells, right? Adult stem cells, of course, will be originally from a fertilized egg. The egg gives everything. So you can have adult stem cell. Now the adult stem cell can repair damage. For example, the lining of the intestine. The cells get replaced. It's because of the adult stem cells there. So this is the natural cause. It's just the differentiation. So this repairing involves stem cells, involves adult stem cell. But pay attention to the middle part, the pink part. So fertilized egg can give rise to some adult stem cells. And in this case, also the stem cells will give rise to some more specified somatic cells. But sometimes when these pink cells are damaged, maybe you don't have sufficient amount of the adult stem cell to replace them. Imagine that type of situation. You just don't have enough somatic stem cells that can be used to regenerate this damage. Maybe the extent of this damage is very big, or it involves cell types that the adult stem cells cannot easily differentiate into. So sometimes what can happen is a transdifferentiation. So for example, if the liver is damaged and now the liver cells, this is a hypothetical situation, okay? I don't know whether it is happening exactly. So if this liver cell is damaged and now you don't have a big source of adult stem cells to replace the damage, or maybe the fibroblast in liver can become liver cells. So that's called a transdifferentiation. That process may not involve stem cell. So just another somatic cell now somehow, get differentiated toward the liver cell, a different somatic cell type. That can happen. As you will see later on, sometimes the epithelial cells can become bone cells. That's a huge change of cell fate, but naturally that can occur. So that's called transdifferentiation. That means one somatic cell becomes another somatic cell. Sometimes we can also use the same term to describe one adult stem cell become another type of adult stem cell. That's also called transdifferentiation. So that's the somatic cells that hold the parallel position in terms of differentiation. Now one changed to the other. It's called transdifferentiation. Okay, so the transdifferentiation apparently is a kind of a regeneration, right? To replace the damaged cell type, but it doesn't involve stem cell. Okay, the third, now in this third scenario, what happens is, okay, maybe you have some damage in certain tissue. You have this somatic cell damage, but there will be some types of somatic cell left during that pathological process. There will be some cells left, not necessarily the main cells in that organ, but maybe some cells are left, and then maybe that cells left can go through a process called deep differentiation. By the term, you can appreciate deep differentiation means from a somatic cell to stem cell. So the cells now change fate from a somatic cell toward stem cell. They become a stem cell, and then that stem cell can differentiate toward the desired cell types to repair the damage. Okay, so for example, it's still the hypothetical example as happening in liver. If the liver cell get damaged, and now you have some other cells that are not damaged, the cell will first become stem cell, and then the stem cell will differentiate toward liver cell. Okay, the first part there, a somatic cell become a stem cell is called a de-differentiation. Okay, in that process, you can tell the stem cell will still be involved, but that stem cell originally was not there. It's created by a somatic cell type. So we describe the three scenarios of regeneration. Not every scenario will involve stem cells. So remember the three terms: differentiation, de-differentiation, and transdifferentiation. So we describe the process of reprogramming. You can tell the reprogramming can also be termed as de-differentiation, right? It's a somatic cell now become pluripotent stem cell. Of course, that's the ultimate extreme example of differentiation that usually won't occur naturally. It's by some kind of manipulation. The cells cannot go all the way towards the pluripotency without manipulation. If you can demonstrate that in the future in your study, that's going to be eye-opening. That's something usually don't occur. So we have three terms that are very useful in study in the field of regeneration. So feel free to interrupt me anytime if you have questions. If not, I'm going to move on to the second part of today's discussion, and we are going to discuss animals that regenerate using stem cells. In these scenarios, we do have the involvement of stem cells. So I'm going to introduce several very good model system, as well as just to put it into other words, the animals that can do a full recovery after amputation. It's a full scale in regeneration. So usually this happens only in invertebrate. When you have a full regeneration, that won't happen in vertebrate. It's almost impossible for vertebrate to have full regeneration. So as I said, try to memorize the names. And here, the top left is called sponge. Sponges in the sea. And then the phyla here, this is phylogenetics. Okay, the phyla here is called peripheral. The peripheral is the phyla name. The sponge is the name for this species. And then the next one shown on the top right, this is called Nidaria. That's the phyla name. The species is called Hydra. That's the Hydra genus, but this is the Hydra here. And the bottom left is the thing we talked in the previous slide. It's planarian flatworm, and shown as the last one, the bottom right, what you see is a worm. It's a segmented worm or earthworm. Probably everybody have encountered an earthworm in the past, most likely. You know, sometimes you do observe there's a band there in their body. That's because somehow, accidentally, they get amputated, but they can regenerate that part. Yeah, if you cut them, sometimes they can regenerate the whole half of themselves. So basically, all the creatures listed here, they are good model systems, except maybe the last one, the segmented worm. We don't usually use the segmented worm as a model system to study regeneration. I would say the planarian are the most widely used these days to study regeneration. Or the top two had their time in history as important models to study. So the important thing about these several creatures is that they can totally regenerate themselves. We want to learn because these creatures are immortal, they never die. So this study these creatures may be able to provide us some insight into aging as well. So maybe eventually we learn some mechanisms. Those mechanisms will make us to become immortal. So don't never die. That's a good thing. Okay. As you can see, stem cell biology, as in our lecture, can get complicated. It can involve some things you learned elsewhere. And here is a phylogenetic tree, a very overly simplified version of it. But why I put this up here is because I want you to have an idea of how those model systems are related to each other. So in a phylogenetic tree, the one that belongs to the root layer, the root, like in this overly simplified version, the root tier will be the sponge, right? The sponge is the root tier. That means it is the most primitive. So the more branch out in this phylogenetic tree, the higher that species will be in terms of evolution, right? So the sponge will be the lowest species in this simplified tree. They belong to the phyla peripheral, and then Hydra belongs to, this branch that is closely related to sponge, but a little bit higher in terms of evolution. So that's Hydra, and then planarian flatworm is a lot more evolved. The names here says I couldn't remember the name. I can't read the name. It's Latin. It's one kind of a planarian flatworm, but this is still invertebrate. And then as you can see later on, this, no matter it's invertebrate or vertebrate, we have additional terms to describe them in terms of evolution. For example, we use a term bilateral to describe the animals that have left and right symmetry, like us. Your left side and your right side have symmetry, right? But in sponge and hydra, this bilateral symmetry is not there. They don't have bilateral symmetry. In hydra, they have some other symmetry, but it's not bilateral. So the bilateral symmetry is a kind of a phenomenon related to evolution. So generally speaking, when you have bilateral symmetry, that means you're higher in terms of evolution. Okay, so scientists are using these different observations to distinguish the animals, in terms of what they're branching out from the evolution tree. Okay, so on this slide, I'm going to describe to you the sponge, how sponge was used as a model of regeneration. And in sponge, I don't know if we have a sponge. Yes, we do have the sponge. I'm going to show you there. That's the sponge, how the sponge look. You see it in the deep in the sea, the sponge. So here it's a section of the sponge. The sponge have limited types of cells. So maybe only about 10 different kinds of cells in their body. So in sponge, you do have two types of stem cells. So perhaps sponge is the prototype of stem cells. In terms of evolution, they have the most primitive stem cells. Later on, maybe all the stem cells are related to this common origin. That's why sponge is important. It is a lower species, but we have the first example of stem cells described in this species. If you go even lower than sponges, and then you don't have any stem cells, not to our knowledge. So you have 10 types of cells in sponge, and this arcucide is the stem cell, and they usually live in a structure like this. This structure is as shown here. You have a secondary stem cell called a coonocyte. It's called coonocyte. This is a very, very weird looking illustration of a coonocyte. So these two sides are the stem cells. So how do you know that they're the stem cells? So basically, if you remove the arcucide, and then the regeneration will not occur. So if you cut that sponge, then at the same time, somehow you remove the arcucide, and then the regeneration will not occur any longer. So that's why they are called stem cells, because they can self-renew, and then if you create a huge damage in the sponge, those cells will be able to give rise to the whole structure again, the whole individual again. And why the coonocyte stem cells here as well? That's because the coonocyte are derived from arcucide. So it's known that this coonocyte are derived from arcucide, but sometimes in some situations they can get dedifferentiated toward arcucide. But the coonocyte is the progenitor of sperm. The arcucide is the progenitor of oocyte. So this creature can have sexual reproduction. That means it involves mating. Two individuals, one's male, the other is female, they can mate and then reproduce to have the next offspring. But they can also regenerate through asexual reproduction. That means if you cut them into several pieces, now they can regenerate themselves and then become several individuals. So they are capable of both asexual and sexual reproduction. Okay, the important concept here is this is the first occasion of stem cells that ever get described. That's the origin of all the stem cells in terms of evolution. And then next, I'm going to describe this wonderful creature called Hydra. Both sponge and Hydra, if you observe them in a laboratory or underwater, you won't think they're animals. You think they're plants, but actually they're animals because the animals capture their food from the environment and then digest it in their body, and then that's what animals does. And here Hydra shows a structure, a section of the Hydra. The Hydra have a foot. The foot is usually anchored to some surface. If you culture the Hydra in a laboratory, you provide a surface, the Hydra will just attach by the foot toward that surface. And then the floating part will be the head, will be called the head, or the hypostome. You have an opening. The food can get into the gastric part and then get digested. You only have two cell layers. One's called the endoderm, The other is called ectoderm. The ectoderm is the outside layer, while the endoderm is the internal layer. Only two layers of cells will be the body wall of this hydra. And in 1710, this famous scientist Abraham Crumley was the first one who described the regeneration power of hydra. And therefore, someone think that he's the first stem cell biologist because he basically described that some species can be immortal. They can completely regenerate themselves. Of course, we don't have his study here. What's shown here to the part to the right is the regeneration by modern scientists. Basically, you cut in the middle, and then the top, the head part, can regain the food and not get re-anchored because the top now is floating, right? After your amputation, but sooner, in a few days, they should be able to regenerate the food and get them anchored again. While the bottom part is still anchored, but they are going to regrow the head, and now they have a mouth. They can take food into their gastric region. So that's what happened. This is not this doesn't have bilateral symmetry. The reason is because if you cut in the middle, you can tell, well, the left and right are not exactly symmetrical. But if you cut from the top, like you're cutting a pizza, imagine that you cut from the top like you're cutting pizza, and now the resulting pieces are quite similar to each other, right? If you cut from the top like you're cutting a pizza, and now all the parts are quite similar to each other. Therefore, this is called a radial symmetry. So this is not bilateral symmetry. This provides a division of the species in terms of evolution. So the hydra later on was known to have three stem cell types. Let me go back to this slide. So usually the stem cells is located between the cells, between the somatic cells. In between somatic cells, like shown here, in the ectoderm, you have cells in round shape. They are located there. Between the somatic cells, you found stem cells. So here is a magnified look of that. So you have the endoderm as one layer, the ectoderm as another layer, and then some of the cells in between the somatic cells are called the stem cell of the ectoderm, and some cells in the endoderm also have stem cells. You have ectoderm stem cells, endoderm stem cells, but some cells located in between those ectoderm cells are called interstitial stem cells or eye cells. So these ectoderm stem cells, they're stem cells, they can self-renew and differentiate, but they're monopotent. They can only give rise to the ectoderm layer, no other cell types. And the endoderm stem cells are also monopotent. They can only give rise to the endoderm cells. However, the interstitial stem cells can give rise to the other cell types throughout the body of this creature. So they can become gland cells, nerve cells, and the nematocyte. So all the cell types throughout the body of that creature. But this eye cell, the interstitial stem cell, can also give rise to the germ cells, the gametes. Therefore, this animal can also reproduce through two ways. One is asexual, if you cut them, or sexual, that's a mating between the male and the females. Okay, so stem cells are involved here, again, in this creature. Then later on, modern scientists, of course, were able to label the three types of stem cells using transgenics. We described that, so you can put a green fluorescence under the control of the promoter of a stem cell specific gene. For example, in this case, they used the EGFP under the regulation of CN-NOS1. So that's a I cell specific gene and therefore I cell specific promoter. You put the EGFP in that locus, and now you can label the interstitial stem cell. And similarly, you can label the other two stem cell types as well by different colors. Okay, and now you can label the cell, you can capture those cells, and you put the cells through sequencing, messenger RNA sequencing. And now you can study their gene expression signature in the 3 cell types. That's exactly what they did. And now they captured the gene expression signature. They know what genes are specifically expressed in each stem cell types. Here you have this diagram that shows the genes specifically expressed in each stem cell types as well as the overlap. Of course, in the center of this diagram, what you will find is the cells that are commonly expressed in the 3 stem cell types. Here we have a list of 5 genes that are common for the 3 stem cell types. So you probably haven't encountered FOXO, but I hope you have already heard something about TCF. TCF is the downstream effector for the Wnt beta-catenin pathway. We learned that Wnt beta-catenin pathway is a major regulator of modern stem cells. As you can see here again, they happened very early in evolution. They're already used in regulating stem cells in lower species. So studying the lower species not only will provide a much more powerful model systems, but also give some insight into their evolution and their mechanisms, their molecular mechanisms in us, in humans. Okay, I guess I will stop here for today's lecture. In the next lecture, I'm going to continue to discuss the rest of the content. I have 24 slides. That's because that was intentionally done this way. I will put them together so you have one place to have all the information about regeneration. Next lecture is about planaria and amphibians. How we're going to describe maybe even more details about regeneration. So if you have questions, please stay behind and ask me. Otherwise, you are free to leave now. Thank you.
Thank you.